Part 4 of our survey of biological macromolecules would be proteins. So proteins are one of the four categories of, or major categories of biological macromolecules. Now, proteins, even though they can serve as an energy source, their primary purpose is body structure and function. Most of the body is made of water and a lot of the rest is made of protein. Refer to your notes and your textbook for examples of proteins in the body and elsewhere. Now, all proteins are made of amino acids. They are large, complex structures made of amino acids. Now, every amino acid has this basic structure. You've got this carboxyl group on one side, the amino group on the other, a carbon in the middle, and something else attached to it, which I refer to as R. Now, there are 20 kinds of amino acids, and they all differ at this location of the R group. There are diagrams in your textbook of the 20 kinds of amino acids. Now, amino acids are the monomers of proteins. They are joined by dehydration synthesis into polymers of proteins, and the covalent bond that connects each of them is referred to as a peptide bond. I did not draw that here. Now, every protein has the following four levels of protein structure. They have a hierarchy of structure. The primary structure, every protein has one. It is the amino acid sequence, which ones you have and in what order. This, of course, is genetically determined. Your genes code for the assembly of these amino acids. Now, the secondary structure is taking this chain of amino acids and coiling it up either into an alpha helix or a beta pleated sheet according to hydrogen bonds between carboxyl and amino groups in the chain. The tertiary structure causes more wadding or coiling or folding into a three-dimensional polypeptide, and it's due to the interaction of the R groups that may attract or repel or have various bonds with each other, forming a three-dimensional shape. Now, the quaternary structure is when two or more polypeptides join into the full-fledged functional protein. So, for example, hemoglobin, it is made of 4 subunits. It is known as a tetrameric protein. Now, proteins have a definite shape, and shape determines what the protein is good for. Anything that interferes with shape can interfere with function. Denaturation is subjecting a protein to harsh or abnormal conditions, and of course that would affect how the hydrogens bond form, that would affect how the R groups interact, and therefore it disrupts the entire shape of the protein. So in other words, subjecting proteins to an abnormal temperature or an abnormal pH will disrupt shape and deactivate function. And this concludes our overview of proteins.